Les cartes à puce, DNA Microarray. DNA microarray analysis is one of the fastest growing new technologies in the fields of genetic research. Scientists are using DNA microarrays to investigate everything from cancer to pest control. Now you can do your own DNA microarray experiment. Here you will use a DNA microarray to investigate the differences between a healthy cell and a cancer cell. Microarray analysis. The human genome contains approximately 21,000 genes. At any given moment, each of our cells has some combination of these genes turned on and others are turned off. How do scientists figure out which are on and which are off? Scientists can answer this question for any cell, sample or tissue by gene expression profiling using a technique called microarray, pronounced microarray. Microarray analysis. Microarray analysis involves breaking open a cell, isolating its genetic contents, identifying all the genes that are turned on in that particular cell and generating a list of those genes. What is DNA microarray? A DNA microarray allows scientists to perform an experiment 1,000 of genes at the same time. It's big. Each spot on a microarray contains multiple identical strands of DNA. The DNA sequence on each spot is unique. Each spot represents one gene. Thousands of spots are arrayed in orderly rows and columns on a solid surface, usually glass. The precise location and sequence of each spot is recorded in a computer database. Microarray can be the size of a microscope slide or even smaller. This virtual lab is broken up into 3 chapters. Chapter 1 and Chapter 2 will give you some background and review a couple of important concepts that will help you to understand DNA microarray technology. And Chapter 3 is the experiment. Chapter 1, Genomics, a new science. What's the big deal about genomics and what does genomics have to do with DNA microarrays anyway? Chapter 3, Measuring gene expression. How can you tell whether a gene is on or off, and what exactly is a DNA microarray? Chapter 3, the experiment. Use a DNA microarray to answer the question, what's the difference between a healthy cell and a cancer cell? Chapter 1, Genomics is a new science. Way back in the 20th century, a few years ago, scientists came up with many ways to study genes, mapping them, making mutations in them, cloning them, sequencing them, and analyzing the proteins they encode. Typically, scientists applied those kinds of studies on one or maybe a few genes at a time, but living these have a lot of genes. Humans, for example, have around 20,000 genes. It would take a very long time to investigate each human gene one at a time. Wouldn't it be great if scientists could study many genes at the same time? Or even better, what if they could learn about every gene in an organism in one single experiment? That's a bold ambition, but that kind of thinking has transformed 20th century genetics into 21st century genomics. Genomics is a new word for a new science. Genomics is a way to study many genes, thousands of genes, or even every gene in an organism, all at once. One proven tool for this task is the DNA microarray. Chapter 2. Measuring gene expression. To see how powerful genomics can be, let's take a closer look and what makes the various types of cells in your bodies different from one another. Skin cells, muscle cells, pancreatic islet cells produce insulin, erythroblast, future red blood cells, melanocyte, pigment cells. Muscle cells, nucleus, strands of DNA, gene 1, 2, 3, 4. With a few exceptions, every cell in your body contains a copy of each of your 20,000 or so genes. Different genes are turned on or off in different cell types. mRNA. So, how do you know whether a gene is turned on? Well, one way is to determine if it is working. Churning out molecules or messenger RNA, or as molecular biologists put it, if the gene is being expressed. Muscle cell, melanin off, actin on, myosin on, insulin off, mRNA, actin 1, myosin 1, and actin, myosin. For example, you might expect a muscle cell to make mRNA for the various muscle proteins, such as actin and myosin, but muscle cells should make no mRNA for the pigment melanin or the hormone insulin. mRNA. Muscle cell. Pancreatic islet cells produce insulin. Gene expression profile muscle. Gene expression profile islet, etc. actin on, myosin on, insulin, melanin off, cyclin 1 on, ubiquitin on, histone 2b1, gene expression profile islet, actin on, insulin, cyclin 1, ubiquitin 1, histone 2, on, and off for myosin and melanin. Bon, a difference between the two cases. If we could measure the amount of mRNA for every gene that is present in a cell or tissue sample, then we could make a genetic catalog of what sorts of biological processes are going on in those cells. And by comparing such gene expression profile from two different cell types, we could learn what makes those cells different from each other. That's where DNA microarray come in. Using DNA microarray, researchers can pinpoint and all the difference in gene expression between two cell types in a single experiment. So let's run a microarray experiment. Before we begin our experiment, we will need a DNA microarray. Here's how to make one. Sequence to entire genomes of the organism you're studying. Fortunately, this has already been done for the organism we're interested in humans. 2. Use massive amounts of computing power to figure out where all the genes are in your sequence. 3. Do more computer crunching to design primer pairs so that you can use PCR to make copies of every genes. 4. Using robots or a team of overworked technicians, make all the primers and do a PCR reaction to copy every genes in the genome. 5. Run quality control tests on all the genes copies you make to find out which PCR reactions didn't work. 6. Repeat steps 3 and 5 as needed to make copies of all the genes that didn't work the last time around. 7. Separate the double-stranded DNA from each gene copy into single strands. 8. Use robots to place microscopic droplets of each single-stranded DNA sample into odd-ordered rows and columns, and a glass microscope slide in this. Donc this is a microarray. 9. Use more computers to create and manage a database to keep track of all the genes spotted on the microarray. 10. Do more quality control to make sure all the spots contains equal amounts of DNA. Is a DNA microarray the same thing as a gene chip? We won't be making our own DNA microarray today. We just happen to have one already made. It's right here in this lab drawer. Actually, preparing a genome-size microarray is too big of an undertaking for most individual scientists and research labs. They usually purchase their microarrays from biotechnology companies, which are equipped to manufacture them in large quantities. microarray, a portion of the 1,000 spots. Each spot represents one gene. Let's take a close look at your DNA microarray, and microarray is made up of thousands of spots. Each spot contains multiple copies of an unique DNA sequence which corresponds to a single gene. As we'll see a little later on, this will be a handy tool for determining the difference between two cell types. Chapter 3, the experiment, healthy cells, cancer cells. We are going to use the power of genomics to answer a very important question. What's the difference between a healthy cell and a cancer cell? Very important. Healthy cell, cancer cell. How am I to try to answer this question? You could look a healthy cell and cancerous cell under a microscope and compare their appearance. That's one way doctors try to diagnose cancer. But cancer cells don't always look so different from healthy cells. And comparing how cancer cells are normal cells look doesn't really get at why they are different. Why they are different. Gene 1, 2, 3, 4. Cancer, gene 1, 2, 3, 4. So, what does make a cancer cell different from a normal cell? Cancer is basically a disease of genes gone bad. In this case, gene 3. Many genes control the way cells grow, divide, and die. When those genes stop working properly, cell growth can spin out of control, leading to tumor formation and cancer. But different kinds of cancer are caused by different sets of genes. So, to be able to better diagnose, understand, and treat cancer, it is key to know which of the genes is in cancer cells and working abnormally. The experiment microarray 1 collect tissue, isolate DNA, isolate mRNA, make labeled DNA copy, 5 apply DNA, 6 scan microarray, 7 analyze data. Donc, the experiment microarray, in this experiment, we will use a DNA microarray to compare the difference in gene expression levels between cancer cells and healthy cells. The step will follow are at the bottom of the screen. We'll highlight them as we go to track our progress. So far, we have a microarray. We're going to need some more equipment. Sample tubes, scalpels, solvent solution, pipette man, and trip, vortex, microcentrifuge, columns with oligo-DT beads, buffer solution, solvent solution, green and red labeling mix, microarray watching solution, microarray scanner and workstation, pipettes we have already says. Now we can begin to answer our question. What's the difference between the healthy cell and the cancer cell? We'll answer this question by looking at gene expression, or which genes are on and off in cancer cells as compared to healthy cells. We will do this by measuring the types of mRNA found in both types of cells. The first thing we need to do is find some cells. Cancer tissue. We will collect mRNA from melanoma, a kind of skin cancer, and healthy skin cells from the same patient. This way we can look at differences in genes expression in cells that have the exact same genetic blueprint. Of course, only physicians and qualified scientists working with physicians would be permitted to collect and use such tissue sample, but for this virtual lab, we'll put on our virtual doctor's caps. Our patient has arrived. Let's get started. Collection tubes. Scalpel, cancer tissue. We will collect the healthy tissue sample and a cancer tissue sample from our patient. We will need a scalpel and 2 empty sample tubes. Drag the scalpel over to the cancer tissue. I take the scalpel and cancer tissue. Healthy, cancer. We can extract the RNA from both tissue samples by dissolving them in a mixture of various organic solvents. We can separate the DNA, proteins, and other cell components, leaving only RNA in solutions. Press next to continue. Solvent, pipette in the tube, healthy and cancer. Drag the pipette down and add some solvent to the healthy tissue sample. I do that. Now add some solvent to the cancer tissue sample. I do that. I do this. Vortex Genie. Next, we will mix our tissues samples on a vortex. As the vortex mixes the sample, the tissue will dissolve and the RNA will be released. Place the tube containing the healthy tissue sample into the vortex. What I do, healthy, in the vortex. Good. Now place the tube containing the cancer tissue sample onto the vortex. I take the tube on the vortex. I do that. It's finished. What a centrifuge. A centrifuge is an instrument that spins at very high speed and separates materials by size or density. The sample have been placed into the centrifuge. The centrifuge will spin the samples and separate the RNA from the rest of the cell's content. I must click on the lid to close the centrifuge. I do that. Press the green button to turn on centrifuge 1. I do this. So, healthy, tube healthy, tube cancer. We have a portion of DNA. and portions of protein, DNA, and other stuff. Why doesn't the DNA stay in solution? The strands of DNA are much longer than RNA molecule. So I can separate RNA and DNA, protein, and other stuff. I have RNA in the tube. So healthy, cancer, clean collection tubes, micropipette tube. Donc now lower the micropipetter so that its tip enters the RNA containing top chase, blue, of the liquid and transfer it to a clean tube. Donc, micropipette in the tube, micropipetter in the tube, in the tube healthy, then in the tube cancer. So it's finished. We have mRNA, tRNA, rRNA. The samples contain several different types of RNA, messenger, RNA messenger, transfer RNA, tRNA, ribosomal RNA, rRNA, breaks. Donc next to continue. Alors mRNA molecules, but not tRNA or rRNA always end in a sequence of adenines known as a poly-A tail. Only the mRNA reflects gene expression, so we'll save the mRNA and get rid of the others. So I have tRNA, mRNA, rRNA, and poly-A tail only on mRNA and not on tRNA or rRNA. So we will wash our RNA samples of the other columns filled with small bits and will only bind to RNA strand that have a poly-A trail. The other molecules will wash away. So I have closed up of a single beads, poly-TS attached to beads. We have a tube, healthy and cancer, with mRNA, tRNA, rRNA, and we'll put them in the column 1 and 2. Alors, pipette our healthy RNA sample column pipette our healthy RNA sample over columns 1. I do this filtration, tRNA and rRNA over. I have just mRNA, and I do this for column 2 in the column 2 from cancer tube. So I have 2 columns with mRNA only. Our columns now contain strands of mRNA attached to the poly-T bits by their poly-A tails. So we have a poly-A tails, mRNA, and poly-T fixed in the poly-A tail. Now buffer how does the buffer detach the mRNA from the bits? Base pairing between DNA molecules is affected by chemical conditions such as salt concentration and pH. The chemical conditions of this buffer do not allow for hybridization, thus the mRNA separates from the poly-T bits. So I have buffer, pipette, columns, donc two columns with mRNA, healthy and cancer, and we have two tubes. Now we will wash the buffer, a buffer solution over the columns to detach the mRNA from the bits. Drag the pipette over to the healthy column. So I do that. Donc the buffer in the columns. And we have two tubes with healthy or cancer with only mRNA. Next, we'll make a DNA copy of the RNA. As we do that, we'll give the DNA some color. We'll add some labeling mix to our two RNA sample. We'll use green labeling mix for the RNA from healthy cells. From the cancer cell RNA, we'll use a labeling mix containing a red fluorescent dye. So alors why do you have to make a cDNA copy with not just not just a mRNA? When we make a cDNA copy of our mRNA, we incorporate a fluorescent label in your DNA, in your cDNA molecule. This label will allow us to visualize the cDNA later on. Also, DNA is much more stable than RNA. The labeling mix will assemble a complementary DNA strand opposite every RNA molecule. Pipette green labeling mixed into your healthy cell sample tube. I do that. Green labeling mixed. Donc mRNA, poly-A12, poly-T primer, label nucleotide, reverse transcriptase. The labeling mix contains poly-T primers, which bind to the poly-A tails on the mRNA, usually referred to by the scientific community as oligo-DT primers. 2. Reverse transcriptase, transcriptase, an enzyme which will synthesize a strand of complementary DNA, cDNA. 3. Labeled nucleotide, which will be incorporated into the new cDNA molecule. The nucleotides in this tube have a green fluorescent molecule attached to them. Labeled cDNA, reverse transcriptase has assembled the labeled nucleotides into a cDNA molecule, and the mRNA molecule has been degraded. So it was in the healthy tube. Pipette red labeling mix into the cancer cell sample tube. I do this. Microarray. Good work. Our tubes no longer contains RNA, but now contains complementary DNA, cDNA. The healthy cell cDNA molecules are green, and the cancer cell cDNA molecules are red. This is what we will apply to our microarray. For the next, two complementary DNA strands from different sources can pair with one another. The process is called hybridization. For the next step in your experiment, we'll take advantage of a key property of DNA, complementary base pairing, is two complementary DNA strands are mixed together. They will soon find and base pair with each other, reforming a double-stranded DNA molecule. It doesn't matter where they come from. It doesn't matter where they come from. They'll do that even if they were not originally paired. Scientists call this hybridization, and it is the key to how DNA microarray work. Hybridization, very important. So we have same DNA sequence, or same DNA sequence, or different DNA sequence. Stuck to our microarray are little pills of single-stranded DNA molecules. A single spot contains many identical copies of the same gems, and each spot on the microarray represents a different gem. So we have microarray database spot number with gens name. For example, spot 235, for example, gen CAMTA1, or sous 236 for example, and the gen will be named RERE. Our computer database lists which then is contained in each spot. It's very important. Microarray LC cancer. Now let's add the single stranded cDNA we prepared from the healthy and cancer cells to your microarray. Remember that all of the cDNA molecule in your tubes represent human genes, and for every molecule of cDNA there is a matching spot of single-stranded DNA on the microarray. Pipette the healthy cDNA sample onto the microarray. I do this. So we have on the microarray un hybridized labeled cDNA over red cancer cDNA hybridized to this spot, or green and red cDNAs hybridized in this spot, green healthy, of course, cDNAs hybridized to this spot. Let's take a closer look at what's happening on the surface on the slide. Most of the cDNA molecules have hybridized to their complementary DNA strands on the microarray. A few stray cDNA molecules did not hybridize. So now, watching solution. Now we will watch of the extra cDNAs that didn't bind to the slide. Place the microarray into the pan of watching solution. I take microarray and I put it in the watching solution. Donc, now let's analyze our data. To do this, we'll need a microarray scanner. Donc, we have a microarray scanner with the button scan green, merge image in yellow, scan green in the green, yellow it's merge image, and red c'est scan red. Place the microarray into the scanner. Microarray into the scanner. I take the microarray, I put it into the scanner. And first, we'll scan the microarray to see where the green labeled cDNA from the healthy cells bound. To simplify things, we'll examine just a section of the microarray. Okay, I must press the scan green button on the microarray scanner. Alors, the green. Donc, alors, the result, green healthy image, all spots are green. Oh. Many of the spots are green, not all, but many of the spots are green, but not every spot. The dark spots are genes that were not transcribed in the healthy cells. Remember, not every gene gets expressed in every cell type. I must press next to continue. Now we'll scan the microarray to see where the red cDNA molecules from the cancer cells bound. I must press the scan red button. I do this, and I have lots of spots again, just are for the green genes, but the red pattern looks different. It's clear that skin cancer cells and healthy skin cells have different gene expression patterns. But how can we sort through all this data to make sense of it? The microarray scanner can merge to the red image with the green image and display the composite picture. I must press the merge image button, yellow. I see spot red, yellow and green. Here are the red and green spots merge together. Any spot that contains both red and green cDNA shows up a yellow. A yellow spot contains genes that hybridized to both green and red cDNA, which means that gen 1 expressed both in cancer cell and in healthy cell. Genes are shown up as yellow spots probably aren't very interesting to us since their activity doesn't change when the cell becomes cancerous. But look at all the red spots and all the green spots. Donc, how do colorblind people see microarray data? The color of the fluorescent labels on the DNA are interpreted as red and green by the microarray analysis software. If you're colorblind, you tell the software to show the spots as yellow and blue. Press next to continue. Merge image. Red spots show the genes that produced more mRNA in the cancer cell than in healthy cells. They're tuned up in cancer. Green spots show genes whose expression is turned down in cancer cells. You are a researcher interested in studying genes that have an increased level of activity in skin cancer cells. What color spots should you choose to study? Red, for example. I'm sorry, the correct answer is red. Red spots represent genes that are turned up in cancer cells. Press next to continue. Wow, that's a lot of red spots. Have all of these genes gone bad? No. Many of these red genes are normal. They are red on your microarray because they are controlled by a gene that has gone bad. For example, genes 4263 is one of the genes that are turned up in cancer cells. Genes, donc these genes produced a protein product whose role is to turn down the expression of several other genes. That color are the spots that are turned down by these genes. Yellow, for example. Sorry, that's incorrect. Genes 4263 turns down a set of genes which are represented by green spots on your microarray. Good job. Gen 6220, for example, is represented by a yellow spot on your microarray. Bon. Healthy cell, cancer cell, gène. Alors, healthy cell, donc gène 6220, for example, is on mRNA and protein. Cancer cell, gene, donc the same genes on mRNA, no protein mutation. While RNA expression usually indicates protein activity, this is not always the case. In this example, genes 6220, for example, mRNA is made in both healthy and cancer cell, causing it to appear yellow on the microarray. However, in cancer cells, a defect prevents the gene from being translated into protein, because this gene shows up as yellow. The defect cannot be detected using microarray analysis. This is a major limitation of the technology. So how can researchers know if a gene is producing protein? That is a problem. Scientists rely on a technique called protein expression analysis to tell them if the mRNA is being translated into protein. DNA microarray analysis is a power tool that can identify genes that are expressed differently in two different cell types by comparing every single gene in a single experiment. That's pretty impressive. However, as we are seeing, DNA microarray analysis does have limitation. DNA microarray analysis on its own cannot tell you which genes went bad to cause a disease, cure a disease, identify every gene that is behaving inappropriately. So, this virtual biotechnique laboratory will walk you through a DNA microarray experiment. DNA microarrays are rather complex. Before you begin, you should have a solid understanding of the basics of DNA and gene expression. So it was DNA microarray, la puce à ADN. La présentation de la puce est terminée. Microarrays finish.